Hello everyone. I hope you are doing good. Consider your immune system like a missile launching system. Whenever an army decides to launch a missile against an enemy target, then there are few things which are considered. Number 1, the target should be specific. So it is logged. At the second level, the higher authorities they decide whether to destroy the target or not. And once the decision has been made, then at the third level, the local commander who is responsible to launch the attack, he makes sure that all the coordinates are set, communication is set, and the missiles have the required fuel, right? And once everything is in order, then he launches the attack. In the same manner, our immune system has certain signals for the T cell activation. And this model is known as the three signal model. Now, why this model is important and why do T cells activation require three signals, we are going to discuss in today's class. It is going to be a very interesting class, so don't miss it. Okay, so let's start. Now, originally it was a two signal hypothesis for the T cell activation, but now it is understood as a three signals. Okay, now the first signal is the antigen recognition. That is the first signal. Now, what happens in that? The T cell receptor it binds to the peptide antigen and M.Sc on the antigen presenting cell. So you can see here, this is the antigen presenting cell, DC, dendritic cell, and this is our T cell, and here it is the M.Sc, right? And here the red color dot you can see it is the peptide. And this one is the TCR, which is the T cell receptor. So T cell receptor it binds to the MHC peptide complex on the antigen presenting cell, and that serves as a first signal, right? Now this is supported by the CD4 binding of MHC2 if the cells are T helper cells, or CD8 binding to MHC1 complex if the cells are the TC cells. So here it is not shown, but these CD4 and CD8 they make sure that they also help in the recognition of the particular signal. Okay, then once this TCR and this APC they come together and they bind, that provides specificity, but then alone it is not enough to activate the T cells, right? We need some more signals. Now the second signal comes is the co-stimulation. Now in case of the co-stimulation, this signal is provided by the interaction between the CD28 which is present on the T cell. So you can see here this is the signal number 2, and here it is the CD28 which is present on this T cell, and then CD80 or 86 on the antigen presenting cell. So here it is CD86 or 80 which is present on the antigen presenting cell, right? So this is the second signal which is known as co-stimulation. Now this particular signal it confirms that the antigen is dangerous and we need to respond, right? So we have now two signals. The first signal is in the form of TCR and MHC antigen binding, and the second is in the form of CD28 and CD80 or 86 binding. Now comes the third signal which is the cytokines, or this is the differentiation signal. So basically cytokines they are released in the environment and they determine how the T cell differentiates. So when there is a particular T cell, when it binds with the antigen presenting cell, then after receiving these two signals, the cytokines determine what kind of effector cells are going to be differentiated. Okay, so they also drive the proliferation and effector function, and that happens via IL-2 which is one of the most important cytokines. Okay, now this signal answers what kind of response should we mount. So basically once we get all these three signals, the T cells are activated in that particular direction. So this is known as the three signal model of T cell activation. Now we are going to understand a bit in detail. Now if you see the signal 1, so the first thing is that TCR it recognizes peptide MHC complex. So here you can see this is signal 1. where here you will see a red color peptide. This is M.Sc and this is T cell receptor, and they are locked. Okay, and here this is CD4 or CD8, depending on whether the T cell is a T helper cell or T cytotoxic cell, right? Then the second part happens here is that the LCK, it phosphorylates the ITAMs of CD3. So this is CD3, and you can see here this is the LCK molecule, and it is phosphorylating the CD3 ITAMs here. Okay, so here the phosphorylation, the P group you can see here. All right, and then once it phosphorylates, it also recruits the ZAP70. So you can see here the ZAP70 molecule is also recruited here, right? In the third part, the ZAP70, it phosphorylates the tyrosine residues of the LAT. So you can see here this is the LAT, and this ZAP70, it is phosphorylating here, which is the tyrosine residues here of the LAT, and with a delay in the Y132. Okay, and then some additional components are recruited to the LAT signalosome, initiating downstream signaling. So you can see a number of different type of components which are being recruited here, and then the downstream signaling happens. So there is an activation of RAS, MEPK, NF kappa beta, mTOR pathways, cytoskeletal rearrangements, and changes in the transcription and translation. So that happens here, after the signal gets, further going down. Now, what is the final outcome of signal 1? The final outcome of signal 1 is the activation of the transcription factors like NFAT, NF kappa beta, and AP1. And we have seen all these, pathways in detail in our previous discussion. Now, these transcription factors, they control the genes for T cell survival, all right, then proliferation and differentiation. So ultimately we are getting these genes upregulated, the survival, the proliferation, and the differentiation of the T cells here. Now the second signal comes is the co-stimulation. Now co-stimulatory receptors, they activate the T cells. So basically this signal is very important and it is critical, although all three are, but this signal decides whether we are moving ahead or not. Now the first is the CD28, it is one of the the most important signal here. Now what happens here, you can see here this is CD28. So it binds to CD80 or CD86. So you can see here CD28 is binding to CD80 here, right, on the APCs, right, and it enhances the IL-2 production, it enhances the survival through the BCL-XL, right, and proliferation. So that is the function of the CD28, and it activates pathway like PI3K, phosphatidyl inositol 3 kinases. These are a set of enzymes which actually do phosphorylations of certain phosphatidyl inositides, right, and they activate different pathways for the survival and activation. Now second is the inducible T cell co-stimulator. This is ICOS, right. Now this ICOS or inducible T cell co-stimulator, it binds the ligand ICOSL, which is the ICOS ligand, and it maintains activity of the effector and the memory T cells. All right. Now this ICOS is absent on naive T cells. Remember, it is absent on naive T cells. It is only absent. It is present when the T cells are under the proliferation and they get the signal there. They are activated. Now, if signal 1 is present, that means the T cell receptor, it binds to the MHC peptide complex, but there is no co-stimulation. Then in that case, the cell, it undergoes clonal energy. So our T cells, they undergo clonal energy, and clonal energy, that it becomes our T cells, they become functionally inactive, or we call them as a unresponsive, although they are alive cells. All right. So it remember that signal 2 is needed for the activation of the T cell. If it is absent, then even the signal 1 is present, that means the binding is there between the T cell and the antigen presenting cell, but the cells will undergo clonal energy. Okay. Now, why it happens? What is the advantage? The advantage is that prevents the activation against self antigen and some harmless antigens, for example, food or commensals or some the bacteria which are present in your gut, which are helpful, and they help you in the digestion. All right. So this process is really very important, and that controls that no autoimmunity take place. Clear? Now, when we are saying co-stimulation, we also have co-inhibitory receptors, and these co-inhibitory receptors, they turn off the T cells. Now, first is the CTLA4, which is the cytotoxic T lymphocyte associated protein. Now you can see this CTLA4, it competes with CD28 for CD80 or 86, and it has a higher affinity for that. And it acts like a brake on the T cell activation. So basically it doesn't want T cell to be activated, and that's why we call it as a co-inhibitory or co-inhibition receptor. Now second is the programmed cell death protein 1. In short, we call it as a PD1. And this PD1, it binds to PD ligand 1 or PD ligand 2, and that suppresses the T cell activity. It maintains tolerance in tissues, and it regulates the T cell differentiation. Clear? And then we have the BTLA. Now BTLA is B and T lymphocyte attenuator, and it binds to herpes virus entry mediator. Okay, this is a kind of receptor, and the name suggests that this receptor is the entry point of the herpes virus. All right, herpes simplex virus, glycoprotein T. Right, but this particular receptor is also important as it is broadly expressed, and it downregulates the immune responses. All right, so now our research is going on, and a special focus is on this particular mediator. Now, balance of co-stimulatory and co-inhibitory signal, it ensures that the immunity is effective. All right, and then there is a prevention of excessive inflammation and autoimmunity. Now, if this check and balance mechanism will not be there, if co-stimulation or co-inhibition, they will not work as per the requirement, all right, and as a balance, then there will be a problem in our body. We are likely to develop some autoimmune diseases, inflammation, and that will give to the overall illness inside the body. So this is really important for that. Now, nowadays, and it has been used, blocking the CTLA4 and PD1, it has become a part of the cancer treatment, okay, as a part of the immune therapy. So here you can see, here this is the CTLA4, and here it is the PD1 or PDL1, the ligand and the receptor, and these are the monoclonal antibodies, and these monoclonal antibodies, they block the CTLA4 and PD1. Now the T cells, they become active again because these receptors are blocked, so inhibition is not there, so stimulation is there, and once stimulation is there, so our T cells, they become active again, and they attack the tumor cells. So this is the part of the cancer treatment. So after a naive T cell receives signal 1, the signal 1 is that our T cell receptor, it is recognizing the peptide M.Sc complex, and signal 2, which is the co-stimulation, for example CD28B7. Signal 3 is provided by the cytokines. All right, this is the signal 3. Now this signal 3 or the cytokines, they enhance the T cell proliferation, and they promote the survival of the T cells. They also guide the differentiation of T cells into effector cells. And IL-2 is the most important cytokines for the T cell proliferation here. All right, it has a very, very critical role. So basically the TCR and the co-stimulation, they upregulate the IL-2 production, and CD25, which is the alpha chain of the IL-2 receptor. Now activated T cells, they not only produce IL-2, but they also respond strongly to IL-2. All right, so the IL-2 is produced by T cells now, which are the activated T cells, and they are also, responding strongly to this IL-2. So basically that the cells go into the autocrine loop, and because of the autocrine loop, you can see here the IL-2 is produced by the activated T cell, receptors are also produced here, and these IL-2, it is binding to the high affinity receptor on the activated T cell, right? Now that cause a very rapid clonal expansion, we call it as a proliferation. The other name is proliferation. So some cytokines here, apart from the IL-2, because IL-2 now it has gone into the autocrine loop and now it is proliferating the activated T cells, there are some other cytokines, they also determine the development of the effector T cell type. What kind of T cell, what kind of effector T cell is going to be differentiated now. Now such cytokines are known as the polarizing cytokines, and these polarizing cytokines, they are secreted by antigen presenting cells, T cells, and innate lymphocyte cells, ILCs. Okay, and these polarizing cytokines, they differentiate the T cells into Th1 or Th2 or Treg or T17, and different type of the T progeny is here. So the role of the cytokine is to differentiate, and before differentiation to clonally expand the T cells, activated T cells. So you can see how these three signals, when they are in agreement, they decide a very strong immune response, and that immune response is not only very specific, precise against an antigen, but also it prevents the undesired outcome like autoimmunity or inflammation, right? So this is a really beautiful model of the T cell activation, and I hope you have understood it properly. So we learned today the three signal model of T cell activation and the role of co-inhibitory receptors in cancer therapy. All right, for today we are pausing our session here, and we will meet you in the next class. Till then, you take care. Bye bye.